awesome. I love protein.
I know you love protein. I love protein too.
Yeah, protein rocks. So you guys talk about protein? I love protein. I love protein too.
We know you love protein. Protein rocks.
Awesome. Protein rocks.
Protein.
Protein.
My class today we will be talking about water and biomolecules. Okay, so I have an AP test tomorrow, and I guess I'm just making this video so I can help myself review and help myself, review the stuff that I need for the test tomorrow. I don't know, I'm gonna be using my notes, so I don't know if that's cheating or not, but yeah. Okay, let's start with water. All right, so water is a polar molecule. Yeah, it's a polar molecule. What does polar mean? Polar means that it's a molecule that has a charge. And why does it have a charge? I believe it has a charge, if I remember correctly, because it has an unequal share of electrons. Yeah, I'm pretty sure that's it. Now, if we break down water, we'll find two atoms, right? We'll find hydrogen and we'll find hydrogen and oxygen. It has one oxygen, two hydrogens. Hydrogen is positive, oxygen is negative. So basically, the two hydrogens are attracted to the oxygen, and that's the polar covalent bond. And this one water molecule can make 4 hydrogen bonds. What is a hydrogen bond, you ask? Hydrogen bond is a bond. Hydrogen bond is I'm pretty sure it's basically similar to polar covalent bonds, I think. Hydrogen bonding occurs like when one water molecule, like the negative in one water molecule. So the oxygen in one water molecule is attracted to the positive of another water molecule. So the oxygen in one water molecule is going to be attracted to the hydrogen of another water molecule, and they're going to attract like magnets. So yeah, that's a hydrogen bond. And each water molecule can make up to 4 hydrogen bonds. 4. Okay, water also has some properties. Some of the properties of water include cohesion, which it's water attracts to water. So basically, water is attracted to water. Likes attracts like, I don't know. Water likes water, and it's basically similar to hydrogen bonding. I assume, right? It's the same thing. Water attracted to water, so yeah, it's basically just that. And then adhesion is when water is attracted to something else, and water is mainly attracted to other things that are charged. So yeah, so water is attracted to something else that isn't water but that is charged. And then we have what's the other one called? It's Carpenter reaction. No wait, I think it's called Carpenter reaction, and that's when both adhesion and cohesion occur. So an example would be like in a tree. You see a tree, there's water inside of it, and the water, it moves up the tree, and one water molecule is attracted to like the sides of the tree, so like it moves up, and because the water molecule is attracted to another water molecule, it moves it moves the other water molecule that's attracted to this one. So basically, look, this water molecule right here is attracted to adhesion occurs, right? So it's attracted to the sides, so basically gonna keep going and keep going, something like that, right? But this water molecule is attracted to this water molecule, to this water molecule, to this water molecule. Since this one right here is gonna keep going like this, the rest of them will just keep on following. I'm pretty sure that's the basic. I don't know. I have my notes here, but I'm pretty sure that's it. Okay, surface tension. Surface tension is basically like a bunch of hydrogen bonds, all right here, like on the surface. You see, on the surface of the water, there's a lot of hydrogen bonds, and they're much closer together than the ones in the bottom, because the ones on the bottom, there's nothing like the ones on the bottom. On top of them, there's water, so that they're attracted to the water there. But the hydrogen bonds on the surface, there's nothing for them to be attracted over here, so they're more attracted to each other. So that creates surface tension, and when that happens, that means that a paper clip or an ant could like stay on the water without sinking. So it's basically, it's like a shield of sort of that kind of like protects the water a little bit, but it's not that strong, obviously, because I can just put my fist in there and it'll go just fine. But yeah, that's surface tension for you. And then we have high specific heat. Water is it requires a lot of heat to heat it up, and the reason for that is because of the hydrogen bonds. You see the hydrogen bonds, they're very close together, and that means that to try and break that apart, you're gonna need a lot of energy to take it apart. So that's why it requires a lot of energy to like break the hydrogen bonds and to heat up water. And to evaporate it, you probably need even more energy to like truly break them apart and all that, I think. Yeah. All right, moving on. Now we're going to move on to biomolecules, class. Now, what do we know about biomolecules? Well, all I know right now is that there's four of them, and they're basically the main properties of life, I think. Life, they make up all life, I guess. So like, the first one will be carbohydrates. Okay, carbohydrate. carbohydrates are the very first thing that your body uses for energy. So it's basically like it's the first source of energy. It's a very fast source of energy, meaning your body doesn't burn it quickly, I think, but it's the first source of energy. Carbohydrates are made of carbon, oxygen, and hydrogen. All four of them do have carbon, oxygen, and hydrogen, but protein and nucleic acids have a little bit more to it than just that. But the first two, carbohydrates and lipids, only have carbon, oxygen, and hydrogen, I believe. So yeah. Anyways, so carbohydrates, yeah, fast source of energy, first source of energy source, and a lot of them is just sugar, a lot of sugar, I guess. And the monomers are the monomers include simple sugars. Yeah, simple sugar. A monomer is just a building block, I guess, like a Lego, like one of the building blocks. A polymer will be multiple building blocks or multiple monomers. So yeah. Anyways, lipids are the stored like fats in your body. They're like they're like what's it called? Long-term energy storage. There you go. So your body, it like holds them down or something like that. Like it holds them. It like locks them up or whatever. Not okay, bad analogy, I think. But it's basically like it locks them, it like puts them in a cage, and then once it's ready to be released, like once your body uses all the carbohydrates, I believe then your body goes to the lipids, so it releases the lipids from the cage or whatever. I don't know what that's showing up, but yeah. Basically that's the basis of it though. Once your body's done with the carbohydrates, it will then go to lipids, which is the stored fats and all that, which is, you know, why a lot of people are like on low carb diets. That way so their body burns to the carbs quickly and they burns faster to the fat, so they lose more fat. Yeah. The lipids there's saturated lipids and there's unsaturated lipids. Saturated lipids, they have no double bonds. What's a double bond? I'm not really sure. But all you have to know is that like it means that it doesn't bend. The lipid is very straight, but unsaturated, fats or lipids, they have a bend because they have a double bond. And saturated fats, they're solid at room temperature, but unsaturated fats are liquid at room temperature. Yeah, because of the double bond and stuff. Anyways, they don't really have any monomers. It's just fatty acids and glycerol. And you don't even have to have the glycerol. You can just be a fatty acid and you can just be a lipid. So, yeah. Alright, I guess moving on now. Proteins. You know, they help build muscle. And proteins are basically built of amino acids. It's basically the monomer of proteins. Now, okay. They help structural support. They build muscle, obviously. And they help enzymes. What do they help enzymes with again? If I remember correctly. I forgot that one. Okay, but proteins, amino acid, they have, their structure is this basically. They have a carboxyl, there you go, and a amine group. So the carboxyl is on this side, the amine is on this side, and they also have an R group. In the center is a carbon, and then carboxyl, and then amine, and then R variable. Every amino acid has everything except the R variable. The R variable is different in every amino acid, which there's 20 of them. In each, the R group determines what amino acid it is. I don't really know any amino acids to be honest with you. I don't remember their names. I just know that the R group determines like which amino acid is gonna be. So yeah, amino acids are they're bonded by peptide bonds. It's a peptide bond. And then yeah, it basically just builds protein. So yeah, and there's like 4 structures. The first structure is the primary structure. And this one, yeah, is basically just like a single string of amino acids bonded by a peptide bond. That's it. It's a very simple structure. It's very simple, not that complex. The second one is a secondary structure. In this one, there's either a helix or a pleated sheet. And basically, this one occurs by hydrogen bonds. They're holded by hydrogen bonds. It's like the first structure, primary structure, it like folds into the secondary structure, like a fold occurs, and they're bonded by hydrogen bonds. Yeah, okay. The tertiary structure, which is the third one. Oh, my fault. Never mind. The secondary structure is not the folded one. I think tertiary structure is the one that is folded. It's basically the secondary structure folded, and that makes the tertiary structure. I think so. Yeah. So the third one is basically the secondary structure folded, and the third one is determined by the R group. Hydrogen bonds, ionic bonds, and group bonds from which direction? Yeah, okay. Determined by the R group. So there you go. And then the fourth one, which is a quadratic structure. This one is like 2 different things. This one is one here and one here. It's not a singular one like the first 3. It's like 2, and it's basically interaction between 2 or more peptide strands or peptide strands. 2 or more peptide strands. Okay, that's protein for you. And then we have nucleic acids. Nucleic acids are what stores or transfers generic information, and they're made of 3 groups. We got sugars, we got phosphate, and we got nucleotide. Not nucleotide. Crap. What's the third one called? It's not nitrogen. No, it's not nitrogen. It's nucleic. No, wait. Sugar, phosphate. I don't remember. What's the other one? Nitrogen base. There it is. Nitrogen base. Nitrogen base. I don't know how I forgot. Nitrogen base. Yeah, so it's made up with 3 groups: the sugar, phosphate, and nitrogen bases. And basically, these ones are basically just they store genetic information and transfer genetic information. Basically, DNA and RNA. And the monomers are nucleic acids. No, nucleotides. Nucleotides. Or it's the monomer. DNA stores genetic information and RNA transfers information. So yeah, that's all I have in the notes. So let me see. Information carries the information that tells how to build the protein. So there we go. There's something there. Each nucleotide has a sugar phosphate group, and a nitrogen base. Okay, yeah, and the sugar for DNA is deoxyribose, while the sugar for the RNA is ribose. Okay. We didn't really go that much into nucleic acids, so even though I learned this in freshman year, I don't really remember a lot of this phase that was 3 years ago. I don't really remember. Like, once I finish a year, I don't remember what I learned that year. I just focus on that for that year, and then I'm moving on to the next year. So yeah, that's basically it. Oh, yeah, I forgot to say one thing about water. Water in its solid state, so when it's ice, is less dense than water at its liquid state, which it breaks a universal law, basically. Which, I mean, I wouldn't say it's a universal law, but it kind of is, because most of the time, a solid is more dense than a liquid. But water is one of the few, if not, I don't know, the only molecule who breaks this, as water is more dense in its liquid state than it is at its solid state. The reason for that is because water, the hydrogen bonds in water, when they're ice, they set apart more. They're not close together like you would normally have, I believe. Like a normal solid, I believe they're more close together, the molecules. While in water, they're more spread apart and they're frozen a bit. So they're, spread apart, I believe. They're more spread apart, I believe. Yeah. Which causes ice to be less dense than water, which allows ice to float on water. And if ice didn't float, we wouldn't be here right now, because it would just be a layer of ice on top of a layer of ice on top of a layer of ice, and the Earth would just be an icing planet. Which I think is a perfect example of like the millions of reasons. Which I think is a perfect example of like, just how lucky we are to be here. Thank the Lord, because this is just one out of millions, if not I don't know how many reasons, or not reasons, of ways of like, if this one thing didn't happen, we wouldn't be here. Like if gravity was stronger, if gravity was weaker, we wouldn't be here. If ice didn't float on water, we wouldn't be here. And there's countless of many more things similar to that. So yeah, I believe that is it. Well, actually, there's 2 more things to cover. Alright, the last thing is dehydration synthesis. And dehydration synthesis builds bonds, and that's basically when you remove water from what do you remove water from? I don't know. You remove water from, I believe I think it could be anything, but I believe it's an amino acid, or like amino acids. I don't really recall that. All I remember is that when you remove water, it builds the bonds, or like it builds the things to come together, I think. And then hydrolysis is when it breaks apart the bond. So you put in water and it's okay. I think I got it. Dehydration is you have one molecule here, or one thing here, and when you put in water to it. No, wait. No, I got it. Wait, hold on, wait. Okay, I got it. You have one thing here, and when you have one thing here, and you take away like an H from here or an OH from here, and you take away that, so you took away water. It allows these things to come together, I think. And hydrolysis is basically you have one thing like this, and you put in water to it. So I put in water, and it breaks apart. So yeah, breaks bonds. It breaks the bond. Hydrolysis, dehydration. It builds the bonds when you add water to it. I believe I got it. There we go. I don't really know. I hopefully I got this tomorrow. It's just AP biology, I guess. It doesn't really feel like AP biology. It feels more like basic biology. It feels a lot like freshman year biology, just a little bit more detailed. I don't know. I'll admit I was expecting it to be a little bit harder, but I can't really be saying that right now because I haven't even taken the test. But yeah, I don't know. I thought it was gonna be like my brain can't comprehend this. But yeah, I think I got the gist of it. Thank you for watching, and in the name of the Father and the Son and the Holy Spirit. Amen. Hey, bro.